Samantha Black, Editor-in-Chief of ScienceBoard, and today I have the great honor of being with Dr. Tal Zaks, who was previously with Moderna as Chief Medical Officer and now is Venture Partner at OrbiMed. Thanks for joining us today, Dr. Zaks.
Yeah, so what we try to do was start with what is different and what is the same when we look at our experience with cancer vaccines, trying to leverage them for the benefit of cancer patients, and what we've been able to do for COVID, for the prevention of COVID. These are almost as diametrically opposite as you could think. One is an infectious disease, the other is cancer. In one case, we're preventing illness, in the other, we're trying to treat it. But the common approach that an mRNA vaccine enables us actually enables us now to start to have a deeper understanding of how the immune system functions and using the lens of an mRNA vaccine in which to study it. And that's what we tried to cover in the session.
Awesome. And can you give a little bit of detail on what the different speakers covered? Because I thought it was really interesting, the different perspectives that they brought to the table.
Well, I think two of the speakers covered our experience with the cancer vaccines as a modality and then extending it into COVID. I think Dr. Wu covered very nicely where we go from here in terms of what the learnings have been and how do we now apply them to improve on what we do with cancer vaccines. And then there was a fascinating talk by Dr. Solomon, a relatively young investigator who's founded a company to actually use modern tools of computation to understand the immune system. In my generation, we used to talk about CD8, CD4s, T helper 1, T helper 2, everything was binary. If you apply modern computational tool, you start to understand the behavior of the cells in our body in a much more nuanced way, in a much more precise way. And instead of looking at populations of cells as a whole, applying single cell analyses to some of these understanding, we're starting to learn about how the immune system functions over time in a much more granular fashion. And the hope is not just in this talk, but we've heard it throughout the conference by others, is that leveraging modern computational tools for single cell analysis of the immune system will lead to novel insights that will enable us to do a better job in the future. Yeah, so the question of burnout is one that comes up frequent because it was stressful. I think what helped me through it was, frankly, the fact that this was a team-based approach. This was a monumental collaboration, both between the people inside Moderna that stepped up to the plate, but all of our colleagues, whether they're at the NIH, the FDA, investigators out there. And I think that sense of mission and purpose and camaraderie around the shared goal is one that really kept us all focused and healthy for a very long time. I have to say that personally for me, going through it was, I think, the most intense experience of my life. I don't like using military analogies, but in this case, it felt like it. And I think there also are some learnings in terms of then making sure you take the time to decompress afterwards. I think we've done it internally with our teams. Once we got to the place we needed to in terms of authorization, I can tell you that I personally, this was, I knew that at the end of this was I will have my time to decompress, if you will, personally. And I think between that and the teamwork that enabled this, we did well. Well, as I step back and look at the field has gone through a number of phenomenal successes in the past decade, right? We've got the checkpoint inhibitors, we have adoptive T-cell therapy, and we're talking about cure. Cure has become a word that is real, that we strive for. That being said, I think to expand on the ability of these modalities to bring cure to more people, there are a couple of things that we still need to be better at. The first is better translation from preclinical models into the clinic. The immune system is very complex, and I think the translatability of the preclinical studies in the era of immuno-oncology has not been as robust to inform us on clinical development. So personally, I think that's been a little bit disappointing, and I think the answer to that is going to be to both develop more models, but also to better understand the human immune system. And I think the other challenge, which I sort of started with, is to really better understand how the immune system functions. I think the analysis that we saw at this talk about looking at exhausted T cells versus novel T cells, do we need to have old T cells that we're now rearming, or do we really need new T cells to come to the fore? Very fundamental questions. We hadn't had the tools to be able to answer them in the past. Now those tools are emerging, and with that, I think I'm optimistic that our understanding of the human immunology will take a step change in the future, and hopefully will bring it with it a step change in our ability to bring some of these curative intent therapies to a wider population. So I think there are three elements here. The first is single cell analyses and the application of machine learning tools to that. I think that is going to be transformative. I think it is bringing some of these tools to clinical trials, to understanding clinical trials. What we used to call translational medicine, I think, is very much still a critical element. And I think the other element here is patient participation. I think it's really critical that we are out there talking to patients, that patients understand what it is we do, that they have realistic expectations, and that they continue to contribute as they have been to our ability to analyze this. If we can't get tissue from cancer patients to do these kinds of analyses, then we can study mice all day long. We won't get the right answers. So it's continuing to partner with patient advocacy groups and patients to understand why this work is so critical, so that we're able to apply these tools to study human disease and human immunology to do better. So what's next? For me, one of the lessons from COVID, as we were in the midst of it, I realized at some point that every Saturday I have to disconnect. And once I disconnected for that Saturday, not for religious purposes, just to let my brain rest, at the end of the day, it kind of clicked for me, oh, this is what I need to be working on next week. And to take that to the next level, leaving Moderna, I've given myself a period of sort of disconnecting and looking at the world of translatable science from a very different approach. It requires, the success that we had at Moderna really is the coming together of various disciplines, engineering, medicine, but also finance. And the ability to fund innovation is critical. And without it, we won't see these kinds of approaches brought to bear. So I've done something perhaps a little bit out of character for myself, which is I've taken a period of time and said, this time I actually want to spend on the finance side. So I've joined a very collaborative venture capital group, OrbiMed. And the reason for me is I found a bunch of really smart people who are in charge of directing investments to the things I care about, which is translatable science. And so I'm going to be spending a period of time with them to learn how they think and hopefully help direct investments as I learn where the current frontier of science is actually moving to.
Awesome. Well, Dr. Zaks, thank you so much for joining us, and we look forward to seeing what you do at OrbiMed.
It's been a real pleasure. Thanks for having me.